The house-dust-mite, often abbreviated as HDM, a common guest in human habitation living from particles of organic matters such as flakes of shed human skin. While these mites are less than half a millimetre long and about a quarter of a millimetre wide, their impact on human beings is considerable. House dust mites produce allergens that are among the most common triggers of allergic asthma. Up to 15%, which is equivalent to 150 million people in the Western world, currently suffer from allergy to HDM. The allergic reactions to HDM range from mild forms like itchiness and running nose to severe forms like allergic asthma and atopic eczema, to life-threatening reactions. Indeed, more than 12 million people in the Western world suffer from HDM-induced allergic asthma, and as yet an incurable disease with high morbidity and high medical need. Patients often suffer from various allergic reactions. The allergy occurs because the immune system of allergic individuals misinterprets a harmless environmental substance as a disease agent and begins producing antibodies of IgE class against it. The way allergic reactions occurs has been subject to worldwide research. The antigens enter the body. An antigen-presenting cell takes up the antigen and presents its epitopes onto its surface through a class II receptor. The activated antigen-presenting cell then migrates to the nearest lymph node. Here it activates T cells that recognize the antigen. Now the decisive step of an allergic reaction happens. The antigen-presenting cell will instruct the T cells to differentiate into either Th1 or Th2 cells. If for whatever reason the responding T cells are turned into Th2 cells, this person will become allergic to the antigen, which is now called an allergen. At the same time also, B cells recognize the antigen and present epitopes on their class II receptors. In case these epitopes are recognized by Th1 cells, the B cells will be instructed to produce IgG antibodies. But in the case of the Th2 cells becoming activated, the B cells will start to produce IgE antibodies. The B cells will be activated and differentiate into plasma cells that actively start synthesizing IgE antibodies specific to the allergen. This IgE binds with very high affinity to effector cells such as basophils in the bloodstream and to mast cells in the tissue. At the same time, IgE also binds to the IgE receptors on antigen-presenting cells. The next time allergens enter the body, these basophils and mast cells, through the IgE on their surface, will bind the allergen, which leads to release of histamine, prostaglandins and leukotrienes, thus causing inflammation of the surrounding tissues, resulting in allergic symptoms. The consequence of an allergen-specific receptor on antigen-presenting cells is that now the threshold for responding to allergen is decreased by a factor of 1,000 or more, which leads to an amplification loop, producing more and more Th2 cells, resulting in more and more IgE. In addition, the presence of IgE receptors on B cells has been proposed as one of the reasons why allergic individuals start to be allergic for one allergen, but soon become allergic to many different and unrelated allergens. So far, medicine has not been able to cure allergies, but only to mitigate the symptoms. Most treatments try to provide air for the patient by using bronchodilators in combination with corticosteroids to inhibit the inflammation.
When a patient with allergic asthma breathes air containing the allergen that he's allergic to, the lungs will close. Modern medication is trying to open up the lungs while at the same time preventing the inflammation that comes along. However, these treatments are not completely without side effects, meaning you need more and more of the medication. Finally, the drugs won't work at all. In addition, if you stop this medication, the symptoms will be back, full-blown.
Recently, new medicaments to help patients suffering from allergic asthma have been introduced. They explore a new way to treat the allergic reaction by neutralizing the IgE. While it is a step forward from the antihistamines, it still has some shortfalls.
So for example, the patient needs to apply the drug every 2 to 3 weeks, and this while injecting enormous amounts of proteins. And of course he needs to do that for his entire life. So you can imagine that this is a very cost-intensive therapy. And also we need to take into consideration that this is only valid for patients with low IgE titers. So those people who suffer most from allergies are not to be treated with this kind of product.
The main shortfall, however, is that once again the drug deals with the allergic symptoms, not the cause. Are allergies incurable? No. Dr. Geert Muller, CSO of S-TIR, has come across a solution that can revolutionize medicine. Geert Muller is an immunologist with a solid track record in international pharmaceutical and biotech companies, 25 years of research, co-inventor of 19 patent applications, co-author of over 80 peer-reviewed publications. His approach is not to treat allergic symptoms, but to intervene at the very start of the allergic cascade. This is at the moment when the T cells react to the presented allergens and receive the information to differentiate into either Th1 cells or Th2 cells. But how to achieve this? What no one has achieved so far. S-Target's answer to that challenge is a drug called SG100.
SG100 is a drug based on a completely new concept. It's based on the capacity of plasmacytoid dendritic cells to produce a cocktail of cytokines that will redirect Th2 cells to become normal, healthy Th1 cells.
Once the balance between Th1 and Th2 cells specific for the allergen is restored to normal, future natural contacts with the allergen will result in a healthy immune response, characterized by the production of protecting IgG molecules instead of allergy-provoking IgE molecules. The allergy is cured. But how does this work in detail?
S-Target therapies are based on our proprietary 2-module S-TIR platform, whereas S-TIR stands for Specific T-Cell Immunity Remodeler.
S-TIR vaccines are comprised of active ingredients that work together in a 2-module system. A proprietary generic module, warhead, and a proprietary disease-specific module, immunogen, linked by high-affinity connectors.
The warhead ensures delivery of the drug to plasmacytoid dendritic cells. The immunogen, part of the drug, contains disease-specific information, which will now be presented by these plasmacytoid dendritic cells to Th2 cells. But through the stimulation of the warhead, these Th2 cells will be converted into healthy, normal Th1 cells.
S-Target's SG100 vaccine operates in 5 steps. The vaccine binds in a specific way to plasmacytoid dendritic cells, PDCs. This triggers internalization of the vaccine and transports it into lysosomes, which in PDCs leads to the presentation of allergen fragments and to the specific activation of the PDC, which triggers PDCs to the secretion of T-cell modulating cytokines, which remodel allergic HDM-specific Th2 cells into healthy Th1 cells, triggering normal immune response to future natural HDM exposure. But how does this model work in practice? Together with Christoph Langer, CEO of S-Target, a biotechnologist and consultant, and former managing director at an international big pharma company, Geert Muller managed to obtain research funding to proceed with the next steps of the development. The market potential of SG100 is enormous. 24 million people suffer from allergic asthma, half of them due to HDM. Based on the revenue and treatment figures of SG100's main competitive product, a very conservative calculation leads to a sales forecast of more than €1 billion for SG100 alone within the first 5 years after launch. SG100 is specifically designed for the human immune system. It therefore only works in humans and the closely related non-human primates. The step from phase 1 to phase 2 in the development of a new drug is often critical. Up to 70% of all drugs fail in phase 2. This is due to the fact that for most drugs, in vivo proof of concept is obtained in a species that has a totally different immune system, such as the mouse, and the data obtained are not predictive for what will happen in the human patients. S-Target strongly reduces this risk of failure in Phase 2 by basing the in vivo POC on a scientifically approved disease model for allergic asthma in cynomolgus monkeys. The clinical symptoms observed in allergic animals when challenged with HDM are practically indistinguishable from what is known from human patients. Cynomolgus monkeys have an immune system that has a 97% homology to the human immune system. Gert Mutter and Christoph Langer are already working on the next step towards developing the full potential of the S-TIR platform. The warhead, designed to find and activate the right target cell, can be combined with any possible immunogen. In practice, this means that any kind of allergy can be treated and cured, just the same way as HDM-induced allergic asthma. This comprises grass pollen, tree pollen, cockroach protein, or cat dander, among others. Market-wise, this means that the number of potential customers of S-Target's products increases by over 20 times, from 12 million to 280 million people, as do the revenues. Still only talking about patients from Europe, the US and Japan, not counting in the rapidly growing number of patients in emerging economies like China or Brazil. Yet still another benefit lies in this revolutionary innovation: personalized medicines. Patients may receive a cocktail consisting of warheads and those immunogens that correspond to their specific allergies. The biggest advantage of S-Target's innovative product, however, is that it is not restricted to allergic individuals, but also people at risk before onset of disease may be treated prophylactically. For example, children from atopic parents have a greater than 75% chance of developing allergies. Treatment with a prophylactic vaccine against HDM, such as SG100, may stop the allergic march. Therefore, contrary to its main competitors and the traditional symptom-treating medications, S-Target's SG100 is a therapeutic vaccine which can be used as a prophylaxis. A vaccine against allergies. This is Gert Mutter's and Christoph Langer's dream: to cure the patients of today and to prevent risk groups from becoming the patients of tomorrow.
Investing in Stargate Therapeutics SG100 will bring benefit to patients, the healthcare system, as well as to the investors.
It's our wish for allergic asthma patients to breathe freely again.